Uh, Good afternoon to all. My name is Samira Aouichat. I am a recent PhD. Today I would like to present our research entitled Melatonin Improves Endoplasmic Reticulum Stress Mediated IL-1 Alpha Pathway in Zucker Diabetic Fatty Rat. The research has been carried out in the Laboratory of Pharmacology of Faculty of Medicine at the University of Granada, where the research focus is on the evaluation of melatonin as a potential therapeutic strategy for the treatment of obesity and type 2 diabetes and their complications. First of all, I would like to give some brief background information about the obesity and diabetes related kidney diseases. Obesity and type 2 diabetes are linked to an increased prevalence of diabetic kidney diseases or diabetic nephropathy, which is an important complication that is emerging as a major cause of morbidity and mortality. The obese and diabetic microenvironment, such as lipotoxicity, glucotoxicity, insulin resistance, hemodynamic changes, hypertension, chronic inflammation, and are known to play a pivotal role in the pathogenesis of diabetic chronic kidney disease, which involve multiple processes, including mitochondrial dysfunction and chronic endoplasmic reticulum stress responses, leading to cell apoptosis and kidney damage. Endoplasmic reticulum stress has been detected in a broad range of kidney diseases, including diabetic nephropathy, and the different degrees of endoplasmic reticulum response could activate inflammation, endoplasmic reticulum stress, anti-inflammatory and could activate inflammation, oxidative stress, apoptosis response, and fibrogenesis. Unfortunately, despite great progress in the control of obesity and type 2 diabetes, there is still no effective target treatment for diabetic kidney disease, given the specific and complex pathogenesis of obesity and diabetes. Currently, the conventional treatment for diabetic kidney disease are control of blood glucose and blood pressure levels. However, even when the glucose level and blood pressure are controlled, the prevalence of kidney disease continues to increase. Hence, there is an urgent medical need for novel pharmacological therapies. Recently, many promising molecules have offered the possibility of slowing disease progression. One such molecule is melatonin. Melatonin was extensively known to possess remarkable anti-obesity and metabolic effects, as well as cell protective properties, including antioxidant, anti-inflammatory, and anti-apoptotic properties. With respect to diabetic kidney disease, there are promising experimental data to suggest that melatonin could improve and even mitigate the progression of the disease, kidney disease, and various mechanisms have been reported to underlie its beneficial effects, more specifically through antioxidant and anti-inflammatory pathways. However, whether melatonin can suppress renal endoplasmic reticulum stress-mediated apoptosis to limit diabetic kidney damage has not been investigated. Therefore, the objective of the present study sought to determine whether melatonin could suppress renal endoplasmic stress and whether could inhibit the related downstream apoptosis response under obese and diabetic conditions. To accomplish this, we used in our study obese diabetic Zucker rat, which is an excellent model of human diabetic-induced kidney disease. This model recapitulates pathological features similar to that seen in human, including progressive insulin resistance, dyslipidemia, hyperglycemia, and all other components of metabolic syndrome. This animal model develops spontaneously diabetic nephropathy around 20 and 20 weeks of age. Its littermate Zucker lean were used as control, which by contrast are non-diabetic and non-obese and exhibit normal metabolic function. At the age of 6 weeks, both Zucker lean and Zucker diabetic rats were divided into 4 groups as follows. 2 groups, Zucker lean and Zucker diabetic fatty rats, received melatonin supplementation in the drinking water, and 2 other groups here, as you can see here, 2 other groups, Zucker lean and Zucker diabetic fatty rats, served as control to their littermates and received vehicle only. Melatonin was received daily at a dose of 10 milligram per kilogram of body weight for 17 weeks. At the end of the experiment, the animal were sacrificed under anesthesia and the kidney of each animal were immediately removed. The renal tissues were used for microscopic analysis and for Western blotting of ER stress response markers and also for ER stress related apoptotic markers. Now I'm going to highlight the result of the study. As you can see here on the figure number 1, the figure shows the effect of melatonin supplementation on the main endoplasmic reticulum stress response markers in the kidney of both lean and diabetic rats as measured by Western blot. From this, we found, as we can see over here, an increased protein levels of GRP78 and phosphoperg, IRI1 alpha and ATF6 in diabetic control diabetic rats as compared to the control lean rats. These results proved that here we can prove that endoplasmic reticulum stress was induced in the control diabetic rats. Interestingly, we found that melatonin supplementation significantly decreased the relative protein levels of GRP78, of IRI1 alpha, of ATF6 in the diabetic group as compared to their counterparts without the supplementation. However, melatonin had no effect, as we can see here, had no effect on the phosphoperg. These clearly indicate that melatonin may exert its renoprotective effect by inhibiting the IRI1 alpha and ITF6 pathways rather than PERG pathway. On the other hand, in the lean group, as we can see over here, melatonin supplementation resulted in a slight but significant decrease in the level of GRP78, but had no effect on the other markers, phosphoperg, phospho-IRI1 alpha, and ATF6. This data possibly reflect the fact that melatonin minimizes oxidative stress that is known to cause accumulation of enfolded proteins that exerts adaptive endoplasmic stress response, which could help maintain cells homeostasis and prevent cell death. This makes sense because, as I have mentioned before, melatonin has been highlighted as a powerful antioxidant. We move to the second figure. The second figure shows the effect of melatonin supplementation on ER stress-mediated apoptotic pathway in Zucker diabetic and Zucker lean rats. Based on our finding showing that melatonin preferentially suppressed the IRI1 alpha and ATF6 pathways rather than PERK, we wanted to investigate the effect of melatonin on the protein expression of the downstream components of the IRI1 alpha pathway, which is known to play a pivotal role in the endoplasmic reticulum stress-induced cell apoptosis. We first examined the expression of GNK, the activated form of GNK. The induction of GNK is regarded as an important element of the switch from the adaptive to pro-apoptotic synapse cascades. As we can see over here, diabetic group without melatonin exhibited a higher level of phospho-GNK as compared to the lean group without melatonin, which clearly indicate that the activation of the apoptotic signaling. It has been reported that the phosphorylation of GNK activate both the pro-apoptotic box and concomitantly inhibited the BCL2 factors. Therefore, it's logical to find an increased level here of apoptotic and anti-apoptotic markers. As we can see, we found an increase of box, BCL2 and cleaved caspase 3 as compared to the control lean. We found also here that the ratio box to BCL2 was significantly higher in the control diabetic group than the control lean group. The ratio box to BCL2 is regarded as a key factor in determining whether cell can enter the apoptosis process. So this result may confirm the activation of the apoptotic pathway. What we appreciate here is that melatonin supplementation resulted in a significant decrease of protein levels of GNK, of box, cleaved caspase, and also significant increase of BCL2. And the ratio box to BCL2 was significantly lower in the supplemented diabetic group than in the control diabetic, to their counterparts without supplementation. But based on this data, we speculate that melatonin protects against diabetic kidney damage by activating pro-survival BCL2 pathway and preventing the upregulation of the pro-apoptotic pathways. The important point to note here is that melatonin, as we can see, restored the GNK effect to those of the control lean rat. We can speculate over here that melatonin intervened with the downstream components of GNK pathway rather than the upstream components of the endoplasmic reticulum stress pathway. Results from our lean rats seem to support our speculation, according to which, as we can see, melatonin increased the level of BCL2 and reduced that of box of cleaved caspase, but had no effect on the level of GNK and IRI1 alpha. This would, on one hand, be maybe in favor of adaptive capacity enhancement, and on the other hand, be explained by the fact that melatonin target components of the BCL signaling in order to suppress the IRI1 alpha pro-apoptotic pathway. What we then wanted to do was to examine whether melatonin can prevent renal structural damage. The histological results showed that diabetic rats exhibited early focal segmental glomerulosclerosis and tubular damage and interstitial fibrosis, as we can see here on this photo image, which are all common features seen in diabetic patients with nephropathy. And for more robust comparison, we used the semi-quantitative scoring method to evaluate renal tissue lesion. From this, we found, as we can see, that glomerulosclerosis index and tubular scarring index scores were significantly higher in the control diabetic rat as compared with the control lean rat. What we appreciate here is that diabetic rats supplemented with melatonin showed no normal structure of renal tissue with lower severity of focal segmental glomerulosclerosis and, interestingly, normal tubules, as we can see, and no signs of fibrosis. We conclude that chronic melatonin supplementation can prevent diabetes kidney disease by suppressing endoplasmic stress and the related pro-apoptotic IRI1 alpha pathway, which reveal its therapeutic potential. The potential renoprotective role of melatonin and its therapeutic value in metabolic disorders, as well as its cell protective effects against